Please welcome to the stage Dr. Amy Jenkins.
For the past 2.5 years, we have been living with and responding to the COVID-19 pandemic. This pandemic has revealed the immense impact that infectious disease threats can have on our health, society, and economy. Hi, good morning. I'm Amy Jenkins. I'm a program manager in the Biological Technologies Office at DARPA, where I focus on rapid response to infectious disease threats and the development and deployment of medical countermeasures. While the COVID-19 response surpassed anything we had deployed previously for an infectious disease threat, in many cases, thanks to new technical innovations, there are still remaining gaps, and it would be unwise not to capitalize on current lessons learned to strengthen our future response capabilities. I tend to think about pandemic response on a timeline, so I'm going to use this graphic here. For example, we can start over here on the left-hand side of this notional graph. This represents the point in time when a threat has yet to be identified, when we are potentially just surveilling for an emerging outbreak. Once a threat emerges, we begin to diagnose portion to determine the disease spread within a human population. From there, we start to transition to the medical countermeasure production and response portion, things like vaccines and deployment of therapeutics. Throughout the entire timeline, we also must focus in fostering the R&D environment to capture and integrate scientific knowledge and deepen our understanding of a pathogen that may be new to us or may be behaving in a way that is new to us. Given the multitude of activities that need to occur during a pandemic response timeline, from biosurveillance to diagnostic development and deployment to vaccine and therapeutic development and deployment, we need to take stock now so that we can respond to the pandemics of the future in a way that ensures that all of these components are interconnected and talking to each other, thereby yielding the most efficient and rapid response. Today, we've invited these distinguished panelists to discuss some of the ways we can leverage what we have learned during the COVID-19 response to develop technologies and approaches to be even more prepared for the next pandemic. These solutions could be based on policy, technical innovation, or even on novel collaborative public-private partnerships within a revamped R&D ecosystem. We look forward to the discussions and would like to welcome the panelists to introduce themselves and provide some opening remarks. So today on our panel, we have Dr. Corinne Bach. She is Director of Pandemic Preparedness and Emergency Response at the Vaccine Research Institute at National Institutes of Health. I've got it. Next, we have Dr. Robert Carnahan. He is a professor and associate director of the Vanderbilt Vaccine Center at Vanderbilt University Medical Center. And then finally, we have Dr. Christopher Houchins, director of the Division of Chemical, Biological, Radiological, and Nuclear Medical Countermeasures at the Biomedical Advanced Research and Development Authority, or BARDA. So given that Chris wins the award for the longest title, we're gonna start down here and let you give some opening remarks.
Great, thank you very much. Really appreciate the opportunity to represent BARDA and be here today with this really distinguished panel. And thank you very much, Amy, for organizing this. So I'm Chris Houchins. I'm the director of BARDA's division of CBRN, Medical Countermeasures at BARDA. And BARDA really has a function in supporting the advanced development of medical countermeasures, vaccines, diagnostics, therapeutics to address some of the biggest issues in CBRN threats, pandemic influenza, and emerging infectious diseases. We do this by working very closely with industry, establishing public-private partnerships, and really facilitating the accelerated development of these much-needed medical countermeasures to be prepared to respond to events in the future. We do this by finding the appropriate authorities and contracting authorities to work with our industry partners. We provide multi-year funding. And really, I would say one of the biggest things that we do is we provide subject matter expertise, technical and scientific expertise to our industry partners as we help to identify risks and mitigate those risks and really kind of work through issues as they crop up. And we also really play a critical role in facilitating partnerships with our other industry partners as well as our other federal partners to identify gaps that our partners are able to fill in this area. So our mission within CBRN is to make sure that we have at least one medical countermeasure, a vaccine, therapeutic diagnostics available to address a range of CBRN threats, whether it's smallpox, anthrax, drug-resistant bacterial infections, radiological threats, nuclear events, burn and blast injuries, whatever it is, a threat that could be engineered or naturally emerging that represents a national health security threat to the United States. And so we really do this by, I would say, in 3 different mechanisms. We invest in the development of medical countermeasures to treat injuries and not necessarily the threat itself, specifically for chemical, radiological, nuclear threats, where you can't really target, you can't really treat, you can't mitigate the threat itself. Like you can't stop ionizing radiation per se, so you have to treat the injuries that are associated with it. We also invest in broad-acting medical countermeasures against our biological threats, against smallpox, anthrax, drug-resistant bacterial infections, whatever it might be, Ebola, obviously. We're looking for opportunities to develop broad-acting medical countermeasures against these threats so that we have those capabilities available should an event occur. An example is we supported the development of vaccines and therapeutics for smallpox, also expanding the label for vaccines and looking at the efficacy of these vaccines and therapeutics for monkeypox so that we have this broad-acting intervention should an event arise as it did in May of this past year. And lastly, we're really interested in expanding the portfolio to develop really innovative, impactful, transformational medical countermeasures to treat the unknown threats, the threat X's. What can we have in place to be able to be prepared to respond immediately should an event occur in the future? So we really have, I would say, a number of strategic priorities that align with these 3 different pillars within CBRN. It's really developing host-based, threat-agnostic MCMs to treat the injury and not the threat itself. And this isn't just for chemical and radiological threats, but it's also for biological threats. So we want to take what we've learned from our CRN, our chemical, radiological, nuclear portfolio and apply it to our biological threats as well. We're looking for opportunities to develop broad-spectrum antivirals with efficacy against multiple viruses. Specifically, if we can, identify central nodes of pathophysiology that multiple families of viruses act through so that we can target those and have broad-acting therapeutics to be able to immediately respond to an evolving event. We also are investing in establishing a comprehensive burn and blast trauma care portfolio. This is, the medical countermeasures that are going to be required to be able to respond to nuclear blast events, but we're also looking for opportunities to get those products into the ERs, into the trauma centers, into the hospitals, so that they can be utilized on a regular basis, through standard practice, so physicians become familiar with them, and these products are already deployed out in the community, out in the healthcare system to be able to respond, not just to, normal everyday injuries that are associated with burn and blast events, but also in the event of a public health emergency or a mass casualty event. We're also looking for opportunities to invest in the post-marketing efforts for approved antibiotics through our Project BioShield authorities. We're really trying to establish a stockpile of really innovative, highly effective antibacterial products that are not only going to help us to be able to be better prepared to respond to secondary bacterial infections following any sort of CBRN event, but also to help the companies really kind of weather this very challenging post-approval phase for antibiotics. And we're also investing in flexible technologies for vaccine manufacturing. Really, we're looking at opportunities to invest in on-demand manufacturing, point-of-care manufacturing. This is a program that was, established a long time ago by DARPA, and we really have tried to pick up the flag and go with it since then. We're looking for those opportunities to be able to have distributed manufacturing, so we really reduce, some of the logistical needs of having to invest in a large stockpile, having a single CMO that's manufacturing a product, and rather have it distributed across the entire U.S. government, and it reduces some of our logistical challenges of getting that product to the patients that need them the most. So really with this portfolio, we're really kind of focused on making sure that we have the appropriate capabilities in place to be able to respond to any sort of CBRN event in the future. And so with that, I just want to say thank you again for inviting me and really look forward to the rest of the panel.
Wonderful. Rob, over to you.
Hi, my name is Rob Carnahan. So I guess the most exciting part about this panel is a chance to get to interact. So I'm just going to introduce myself just well enough so we can get to like what would be relevant questions for me. So in some sense, I guess I'm representing the role of an academic lab or center in the kind of consortiums that we can talk about. We're a relatively large center of about 30 or 40 people, but beyond just our own academic center, our P3 Performer group included both other academic sites and small biotechs and eventually large pharma. So we also played a role at this sort of pulling together public-private partnerships in our response to the pandemic. So anyway, I co-run the Vanderbilt Vaccine Center with James Crow. Just to let you know, the name is a bit of a misnomer. We actually don't do vaccines anymore. It's a historical name. We actually do almost exclusively monoclonal antibodies to viral targets. We think of that activity in two big buckets. One we call AHEAD 100, and one is the P3 rapid response that we did underneath the DARPA funding. We don't think of those as like completely separate. We actually think of AHEAD 100, which is the idea behind that is we've assembled a list of 100 pathogens we think are likely to be the source of future pandemics. And our idea is to get ahead of those and get antibody drugs into late preclinical or ideally even a phase 1 clinical trial now so that they're on the shelf for later. You can see we've had several stories where we can see the value of that. The most recent one is monkeypox, where we had antibodies we discovered a couple years ago in response to African outbreaks to monkeypox, and now those antibodies, which were sitting on the shelf in our lab, are sort of moving towards late preclinical and early clinical stage. So AHEAD 100 can actually be part of a pandemic response. We also saw this in the pandemic with Vir, which took a SARS-1 antibody that they'd already had, and were able to show that it had efficacy against SARS-2. So again, prior work on potential pathogens helps us respond in the moment. Though we're aware that like no amount of probably pre-work is going to hedge every threat. So then building a pipeline to respond, or at least adapt things that we already have to the current outbreaks is also essential. And that's where something like P3 comes in. So we were a performer site. We were the prime for a group of about 5 sites that responded. We began in 2018. Building our pipeline, we went through several capabilities demonstrations. We can talk about the value of those. I think that was a huge part of how we were able to respond in the moment to SARS-CoV-2 was those capabilities demonstrations, which pushed us beyond like the theoretical ideas of how fast we could do things to very measurable and observable outcomes as to how we could respond to those things. So through those capabilities demonstrations, we were able to mount a response in COVID. We discovered antibodies. We actually did the antibody discovery about twice as fast as we'd ever done it prior to that, and we could talk some about that. And then through, honestly, another huge role we can talk about is the role of, I think of it as like a hub and spoke, with government organizations being the hub, really, of making groups like myself, pharma and biotech sort of pull together and align our motivations, so to speak, so that we can move something forward. And Amy was our program officer and was hugely critical in the discoveries we made, being sort of ultimately went to AstraZeneca and were the antibodies that were a part of the Evusheld product, which is now out with an EUA. So we can talk about that as well. So with that, I think that's enough to let you know who I am, and I'll stop there.
Thanks, Rob and Corinne.
Thank you, Amy, and everybody. I'm very honored to be part of this. So I am the Director of Pandemic Preparedness at the Vaccine Research Center. The Vaccine Research Center is a division, intramural division under Dr. Fauci, so that's the easiest way to think about it. And it's also kind of a misnomer because we do vaccines, but also monoclonal antibodies, half and half. So it was created about 20 years ago to fill the gap of having a government-owned, what we like to call biotech organization, because we have end-to-end capabilities. We start at the R&D, we do testing, we do clinical testing, we do manufacturing, we do process development, and in the end, it's very complementary to what BARDA does, because we go all the way to maybe a phase 1 or a phase 2. And then our end goal is to have that medical countermeasure, either a vaccine or a monoclonal antibody, license the technology to someone, a company or a nonprofit organization that can take it all the way to either licensure or application or expanded access or whatever is needed. So one of the things, we have many accomplishments, and I'm very proud of our team, but one of the things that we did is during the pandemic, the coronavirus pandemic, we had the first in human monoclonal antibody and the first in human vaccine, which for us was huge because we had been responded to emergencies for a long time since probably SARS-1, and we had been trying to compress that timeline smaller and smaller and faster. So it took us only 65 days from the design of the vaccine, the Moderna vaccine, to a first in human, and that was a milestone for us, and we hope to get better. So our technology didn't stop there. It was used for many of the vaccines under Operation Warp Speed, and a lot of our scientists were part of Operation Warp Speed with BARDA and DoD and other government colleagues. So the other thing I would like to highlight is that we work with two kind of scenarios, if you want to put it that way. When the medical countermeasure has a commercial interest, we usually license to a pharmaceutical that's interested in moving it forward. I don't know if you heard in the last couple of weeks about Glaxo and the RSV vaccine and the great results there from their phase 3 trial. That was our vaccine that was licensed to Glaxo. But we have a lot more work when the medical countermeasure does not have a commercial interest, and that's where we have to really entice either a small biotech or a nonprofit organization to take it forward, and maybe if it's in BARDA's portfolio, work with BARDA or get any other kind of funding. So that's the line of work that we struggle the most with, and that we really share with DARPA, and we've been great partners working on that. So I think that's all I have to say.
Wonderful, thank you. So I think we'll move on to a couple questions here. You should see a QR code pop up. Please feel free to submit your questions, and while we're waiting for audience questions to come in, we'll tackle a couple things that we've been talking about here. So I think we're gonna hand this over maybe to Chris, but what were some of the initial technical and logistical hurdles in your opinion in the early COVID-19 response, and how has technical or policy changes helped overcome those hurdles?
No, thank you. So it was funny. I'm just kind of reacting to something that Rob said about how U.S. government can really play a role in providing motivation, to other developers and sponsors and partners. And I would say that one of the hurdles that we had to overcome quickly was in the very beginning of the response, we didn't have any dedicated funding to be able to respond to COVID-19. So we had to pull funding out of HIDE, out of other programs, which kind of created a preparedness gap for those other threats, to be honest with you. But thankfully, in March and April, Congress was able to provide us significant funding, tens of billions of dollars, to be able to really accelerate the research and development of promising COVID-19 therapeutics and vaccines and diagnostics. And it wasn't so much the motivation or the funding, but what the funding allowed us to do was, by working with our industry partners, we were able to do a few things. One, we were able to establish a portfolio of products. So we invested in 6 different vaccine candidates with understanding that several of these are going to fail, but we needed 1 or 2 successes. So we made a decision to invest in that portfolio, which is very expensive when you know that you're going to have failures. Number 2, it allowed us to be able to support the multiple activities going on in parallel. So obviously, for a lot of medical countermeasures and products, you're going to complete a phase 3 registrational study before you start commercial scale manufacturing of that. We were able to do those activities at the same time. And start commercial scale manufacturing before we even have the readout from a phase 3 study. Again, very risky because that, if the readout showed that the product didn't work, wasn't efficacious, then all the product that we manufactured really didn't have any value whatsoever. And third, it really allowed us to be able to, provide advance purchase commitments to our industry partners, to the manufacturers, so that those companies knew that once the product got approved, received emergency use authorization from the FDA, that we were able to purchase it. Mm-hmm. I would say the other area that allowed us kind of really to make really significant advances very quickly was in kind of turning to those platforms that we had long supported and been a part of, that allowed us to quickly manufacture and develop and manufacture vaccines and therapeutics. And specifically talking about the mRNA platforms that we've worked very closely with DARPA and NIH on. but also therapeutic platforms such as Regeneron's VelocImmune platform for monoclonal antibodies. So by having those prior investments in those platform capabilities and platform technologies, we're able to turn quickly to those and really start accelerating the development of these much needed medical countermeasures.
Great, yeah, platforms are important and risk is not always a bad thing. I think that's a theme the last 2 days. All right, Rob, you set this up in your opening remarks. So how can we leverage the defense and industrial bioeconomy biotechnology base for immediate pandemic responses in the future?
Yeah, so I'll go back to, I guess I'll start with, I guess this is defense, maybe this could be ARPA-H as well in a similar way, but we have been developing a pipeline to respond with monoclonal antibodies to viral diseases for a long time. One key difference when we became a DARPA performer is, again, like I said, that went from a theoretical, our main output for that is publications, right? So in a publication, one way to get something published is to always look at your data with rose-colored glasses and say, well, we did this faster than last time. So a key difference when you come into a program like DARPA is that you can't just talk about how you did it faster. You literally have a stopwatch that you're starting and stopping. And so you really you're forced into to consider constraints that you don't typically consider, like even sort of ones that people don't think about. So our consortium, for example, literally went from coast to coast. We had performers on the East Coast and the West Coast and 2 performers in the middle. So when we were doing our capabilities demonstrations, like we did a response to Zika, where we were literally held accountable for every day, including Saturdays and Sundays and 24 hours a day, which we wouldn't normally do. But on top of that, we had to consider logistics as part of that. How were we going to get samples shipped, you know, on Saturday afternoon from Seattle to Nashville, and we had to work all that out. And that was a key part of us sort of thinking through how we're going to do a platform response and the push from a group like DARPA and Defense to really think about like the demonstration of what you think you can do in real terms, and then pushing that into like how you ask the next question. Okay, well, I now have solved that problem. You also are pushing timelines that aren't necessarily relevant normally in academics, where a week may not be a big deal. It is a big deal when you're talking about a pandemic response, and so pushing the capabilities where, yeah, really, we do need people here on Saturdays and Sundays, because that makes a difference. Again, and the other thing sort of more globally to this question is defense, a group like DARPA, everyone, I think there's been a lot of popular media coverage of the role the government has played in funding and de-risking from a funding point of view a lot of what happened, and there's no doubt we were all able to take risks. We were even able to take risks not just across sort of our sector, antibody development, but within our lab, we took risks, right? We know people who didn't enjoy the kind of program we were in. I know one in particular, I won't mention them, but they took a big risk in how they were going to do their antibody development sort of program. That risk turned out not to work, and then they were kind of at a dead end, right? That was the end of their response. We were terrified at the beginning that we were going to fail, and so we literally launched 10 different platforms at the same time, like most of which we had never done before, but were kind of related to things we'd done before. One of which included acquiring an instrument that cost a lot of money, and we had it delivered within a week, and within 2 weeks we were doing new work on it. So we were able to take huge risks because of the support we had. But beyond that, it was this idea of like the government performer playing an arbiter role, right? So helping us align. When we were talking with AstraZeneca, we would normally talk to a group like that at the end, kind of near the end of the road, like licensing discussions. We have an antibody, let's talk about your interest in it. Because they were a P3 performer and we were a P3 performer, they were literally in our business. So when we were doing our initial discovery efforts, they were there. They were talking about things they liked, things they didn't like. We were able to sort of tweak things and move things. And ultimately, what we ended up with was discovering a panel of antibodies that we had to choose from. So we had sort of an abundance of riches. Which ones are we going to move forward? Which ones are we going to sort of invest in further? And we were able to have direct conversations with DARPA as the mediator with our industry partner, right? So I think that hub of like really aligning motivations and milestones across groups that have very different outputs, right? So industry output is more of a long-term sustain. They want to build platforms that are not only going to deliver in this pandemic, but they're going to be able to use those to respond to other kinds of things, maybe not even infectious disease. Whereas the strength in our world is like we can build a platform in an instant. We don't care that much because our output is publication. So we don't, if it's not going to be useful in 3 to 5 years, that's really not that important to us. If in the meantime we can get some mileage out of it for publication. So very different ways of worldviews, but having someone in the middle who can kind of help us find wins for both of us was, I think, a key part of like the role of government and defense can play in that.
Wonderful, thanks, Rob. So I think we're going to move on to the more early stage, talk about some R&D things. So maybe we'll start with Corinne. Can you describe some innovative current R&D ecosystem and collaboration between academic and industrial labs that may provide a framework for future response? What are we going to be doing in the R&D space?
So that's a great question. Because VRC is at an inflection point, we've been here for 20 years, we've learned about this business, we learn what we do best, what we don't. And our center was founded on HIV research to try to address HIV research. We're still trying to address HIV research, but it's not lost on us that everything we learn from HIV has been trickled down on other viruses, especially coronavirus. Without HIV, we wouldn't have the coronavirus vaccines we have today. So I think as we look to the next 20 years, we need to take stock of all the things we do well and all the systems we've established for viruses we have, and transfer that to viruses we want to invest in the future, especially with the priority prototype pathogen model that NIAID has published as our strategic framework for addressing pandemic preparedness. And I have to say, I agree with Rob. We do, at the beginning, we looked at the industry or biotech as an end step to our process. But over the last few years, we've started incorporating them more and more, even before that IP is sent to the patent office or anything like that. So they come with us when they see we have an interesting concept, and they start working with us from the beginning, trying to collaborate with us and use their subject matter expertise to make it even better. So some of our collaborators in the industry are with us from the beginning, and the same is true for academic collaborators. I don't know if you have anything to add, Rob?
I guess I'll just, not really. I mean, I guess I think it's exciting that you mentioned the prototype pathogen.
Yeah. Mm-hmm.
I think thinking like that, which I would say merges well with our idea of a HEAD 100, we've solved exactly one sort of coronavirus pandemic, really. We don't know if other ones will follow on. I mean, it wasn't really a novel coronavirus in our words. It was a pseudo-novel coronavirus, and we were very fortunate in that way. Thanks to work at the VRC by Barney Graham and Jason McClellan, we, in antibody space and vaccine space, were able to take all that information and actually respond. So we haven't responded to novel pathogens per se. I won't go down that right now, but like, you know, continuing this idea that we need to attack new pathogens and figure out what makes them tick and how we can respond to those. It may be that we, like in a case I just said of monkeypox and other ones, we get lucky, well, not lucky, but lucky in a sense we've already worked on it and there's some antibodies out there. But even if we don't, we've learned about something close to that, like what the VRC had done with SARS-1, that we were able to pick up and move that information into a new way, a new virus, a new way of thinking. And programs like the JPO's Accelerating Antibodies, I think fits well into like a follow-on with DARPA, where we're going to participate with JPO being in that hub and helping us as an academic participate with industry to get a few viral antibodies into phase 1 clinical trials. Again, ones that would give us more chances to practice, more capabilities demonstrations, more knowledge about specific viral families that we hope will translate to other viruses.
Right. And I think what an important kind of aside here is that oftentimes when we think of pandemic response, we go straight to the things that are deploying the vaccines, manufacturing, because that's the most visible, that's what we need. But laying that R&D background and really understanding these pathogens and being able to study them from a fundamental scientific perspective is critical. But that doesn't mean we can't have innovation there. And so I think that there's a lot of opportunities to think about how innovation can actually help us do very fundamental biology. There's still space there, and I think that's a really exciting realm that we can be in the future of pandemic response. All right, we're going to move on to another question. What are some of the pathways to rapid medical countermeasure development for effective logistics and deployment, and what gaps remain? So Chris, I'll start with you. I think you probably touched on this a little bit at the beginning, but I'd love to hear some more of your thoughts.
Yeah, no, really appreciate this, and it's a great question. So one of the areas that we're really interested in, I think a lot of us are interested in, our platform capabilities, obviously. And we really have I would say, put the mRNA vaccine platform technology through its paces over the past couple of years, and it's really demonstrated tremendous value in terms of being able to deliver effective capabilities to respond rapidly to an emerging event. We want to do the same thing for therapeutics. We want to look for new opportunities to rapidly discover, develop, and deliver effective, safe therapeutics in response to an emerging event. So what does that mean? So, I mean, there have been investments, obviously, that DARPA has made over the past years. Renee touched on this earlier, what Dan Wattendorf did with DNA-based monoclonal antibodies. We want to explore that space a little bit more. We want to explore the ability to rapidly develop nucleic acid-based monoclonal antibodies to be able to respond rapidly to an event. We're looking at other types of technologies too, like siRNA or CRISPR RNA, where we can target the pathogens directly to be able to affect and impact some critical mechanism within that pathogen. We're also looking at threat agnostic approaches, as I indicated before. What are those central nodes of pathophysiology that we can target, or those central nodes of pathogen replication, transmission, egress, infectivity, whatever it is, that we can target it without exactly knowing what the pathogen is, what the viral pathogen is, but it's going to cover a wide range of families of pathogens, for example, so that we can target those directly. I would say, so those are some of the challenges, those areas that we're really interested in. Some of the other areas we're also interested in, I had indicated this before, is point of care, on-demand manufacturing. Having distributed manufacturing of vaccines and therapeutics, across, let's say, the entire United States or across the globe. So we're not focused on, a central CMO to manufacture a particular countermeasure. We have it distributed across the, entire space, I would say, so that one, we're manufacturing and delivering those medical countermeasures closer to the people that need them. So that we're reducing the logistical challenges of having to ship, products across the entire U.S. or the entire globe. Number two, you don't have a single point of failure. You have robustness in the system. So if, a single line of the CMO goes down and that's your only line, then you're offline for a very long time. And really, it also, I would say third, it reduces our dependency on building and maintaining and paying for a large stockpile of medical countermeasures that hopefully we're never going to need. So if you can, identify based upon the sequence of a pathogen, this is what the vaccine needs to look like. And every CVS, every Walgreens has their machine that's able to create a vaccine for that local population. That's an area that's going to have we think a significant impact. And lastly, we're looking at other ways to facilitate the distribution and administration of vaccines and therapeutics, such as needleless patches or microneedle patches that reduce the dependency on ancillary supplies, which was quite a challenge in the beginning of the COVID-19 response, looking for new capability, like oral vaccines, for example, things that improve our CONOPS for any of the medical countermeasures that we're developing.
And given the VRC's kind of interest in taking these products through phase 1, what are your thoughts?
So that's a great question. So I think the short answer to that question is that we were lucky that it was a coronavirus that emerged, because we had so much work done on MERS and SARS-1 that we really knew what to do. We were not as far along as we had wanted to do because, as we mentioned before, these viruses that don't have a commercial market for their medical countermeasures are very hard to advance. So from the early stage development that the VRC space is on, I think we need to make sure that all those virus families that we don't have any knowledge on, and even disease X, which is an unknown virus that might emerge, that we're prepared for that. And the other part of that answer is that if we had at least a phase 1 already, or if we had some product on the shelf, even if it wasn't for SARS-CoV-2, but for at least another coronavirus with a similar platform, I think we could have made it even faster. And I agree with mRNA. We are also at VRC investing in mRNA technology, but we have to remember that we need to tell the synthesis machine what sequence to put in the mRNA. And that takes a lot of R&D for many different viruses. So,
Yeah, great.
And if I could just add on, one of I would say the bright spots of, and I think that there are many of the COVID-19 pandemic and our response, was it has really brought so many different communities across the U.S. government together. I mean, obviously, you know, BARDA and NIH and DARPA and, you know, lots of other organizations, you know, prior to COVID-19, yes, we would get together, we do portfolio reviews, we talk every now and then, but for the first time, we have integrated and worked together in a way that we've never have done before. And I think these are going to be resilient relationships that are going to make us much better prepared to respond to future events.
I agree.
Absolutely. Yeah.
Just like Renee said, her resilient systems, maybe our U.S. government, our ecosystem can be a resilient system as well. We'll add an ARPA-H. Okay, so we got some great audience questions here. So the first one, what lessons from the COVID-19 pandemic moved us towards preparing for the next pandemic threat? And I think we've been identifying some of those. But if a new outbreak was announced next month, what should we do differently? Nobody wants to answer that question. Come on.
Go, please.
You want to start?
So it's going to depend on what outbreak. And every one of those possibilities has a different answer. Who has and what do they have? I have to say we are much better prepared and much better organized than we were before the pandemic. Chris was absolutely right. We knew each other before, but now we're besties, and we just call each other all the time and say, you know, get it together, and I help you, you help me. So yeah, but in the end, it has to depend. But I think that the concept of prototype pathogen is very important, because that takes a problem that is very hard to even address or start to or begin to address, which would be a DARPA problem, into something that we can start chipping a little bit, little by little. So yeah, sorry, Chris, go ahead.
Oh, no, I think you really captured it. I mean, one of the ways that we've been looking at it, and this is not going to happen next week, so please hold it off for a while, the next outbreak. But it really is, I think, looking at pandemic preparedness from a layered approach. You know, as I indicated, you know, I think we want to have, you know, therapies, for example, that are available, you know, 8 o'clock day one when an event occurs, so that we're able to respond to the medical consequences of an emerging event. You know, whether it's, you know, ARDS or sepsis, vascular leakage, whatever it might be. We want to have something that's ready to go, able to treat the injuries, the medical consequences associated with that event. Not going to be necessarily 100% effective at reducing morbidity and mortality, but hopefully it's going to help lessen the impact. The second layer is having more broad-acting, you know, interventions still available right away, you know, at the start of an event. So, you know, these could be, you know, broad-acting small molecules, or, you know, different types of therapies that are, you know, focused on, you know, common mechanisms of viral pathogenesis, for example. Again, not 100% effective at reducing morbidity and mortality, but what it does, it allows us to buy time, allows us to flatten the curve a little bit, reduce the incidence of infectivity, reduce the incidence of mortality, while the definitive medical countermeasures are being developed, which is going to take a little bit more time. You know, the definitive monoclonal antibodies based upon whether it's, you know, prototype pathogens or vaccines to be able to, you know, respond in a much more effective way. You know, but that takes time. But the way that we see it is, you know, we need something available right away. Again, not 100%, but we need to be able to respond quickly while, you know, those more definitive vaccines and therapeutics are being developed.
Yeah. Rob?
Yeah, so I'll go back to this idea of, like, I do think that a lot of friends have been made within the government, and particularly as the pandemic went along, there was a better and better coordination. I think one thing that we should do differently is start to export that cooperation and coordination outside the government. So, for example, one of our key needs to launch our platform is patient samples. We actually need people who've been infected, and finding those patient samples is really complicated. And since there had been no forethought as to how to coordinate finding those samples, we were dealing with institution by institution, which is an academic side. We actually could do that. For example, industry, a lot of them just got locked down. That was not something they had the capabilities to do, and we could go into why that is. But we had an organization we contacted who had access to early patient samples, and they said, sure, we're going to let you have samples. We're forming a standing committee that's going to allow us to navigate the existing rules about when we can provide those to you. And then about 3 months later, they got back to us and said, we have now determined that you can have those samples, right? So by that time, we'd already had our antibodies, and they were in production. So that to say is like they needed some permission ahead of time. We have to think ahead of time, how are we going to give organizations permission? Our institution struggled with biosafety, which is accountable to the NIH. They were very scared about changing flexible biosafety rules in response to a pandemic. They didn't let us do some things which would have been really helpful. But someone would have had to think about that ahead of time. I think the government is the group to coordinate that, like going out to academia and industry and talking about ahead of time. How could we make normal rules still safe, but more flexible so that in a pandemic, we can sort of break the glass on the fire, pull the fire alarm, and let you do some things that are still controlled but aren't normally allowed. And we didn't have that, and that really was a big struggle for us.
I think one final point I want to put on this is that you're hearing that there's a lot of collaboration across the government. We're all besties now. One opportunity that provides for groups that are maybe new to DARPA or get DARPA funding, oftentimes, particularly in the bio space, interactions with DARPA are very early, very fundamental R&D. And because we do have these great relationships with our partners across the government, you often have the opportunity to see your product on a commercial on NPR, right, Rob? So it is a great, I think, feeling to see these products make it all the way through the process. So, all right, let's move on to the next question. So another audience question here: monoclonal antibodies are important for treatment, but they are also critical reagents for antigen detection tests, including rapid tests. Have you also considered partnering with diagnostic test manufacturers in addition to treatment applications? And I'm going to go straight to Rob on this one. Yeah.
Yes and no. So the considerations for doing a diagnostic, so yeah, they're the same, they're antibodies, but oftentimes it's not even the same protein you're targeting necessarily. And so we think our best use case is to not think too much about that. We did partner with some people. Basically, we are 100% focused on what do we think is going to generate a protective antibody and how that's going to translate into in vivo protection. When we discover antibodies, depending on how we do it, there might be ones that are interesting for diagnostic, but we literally we don't have the expertise or bandwidth. We probably could partner with someone to do some of that, but I think it's probably better to have a more fit-for-purpose group that's like from the beginning. And I know one of my friends, Jason Goldstein from the CDC, that's what they do. Like from day one, they're thinking about a very different lens on the problem. How can they develop diagnostic antibodies? What are the key targets? How would they put them through their paces? Because we choose antibodies early, and we're choosing them based on potential therapeutic value, not based on rapid antigen detection criteria. And it's a little difficult to sort of walk and chew gum in that sense at the same time.
So maybe early collaborations may provide some benefit there. Great. Any additional questions? Any additional comments related to the diagnostics? We're largely not as focused on diagnostics. All right, let's do a final question here before we wrap up. Can we do a war game pandemics and their response like they do in the military space? And I think this is probably one everybody's going to jump at here, so we'll start here with you, Corinne.
So we had done, I don't know if it was a modeling computer, but we had done before roundtables, right? Where we
tabletop exercise.
Tabletop, yeah, exactly. I didn't have the term. So we had done that at the very least for influenza and for other potential. We thought it was going to be influenza. Influenza was very likely. So we had brought everybody from the government that would address or we would work on a pandemic situation and try to do that. My personal opinion or my personal experience was that it was a lot bigger than that and a lot more complicated than we had anticipated. But I have to say, the government rocks. I mean, it's everybody, I'm sorry, I'm just so proud of my colleagues. Everybody came together, and this is hundreds and hundreds of scientists and biotechnologists coming together from HHS and DOD and saying, we got this. And everybody worked on their lane to make it happen.
Yeah, no, I would agree with that. I think one of the earliest things that the government did was, each of the different agencies and organizations in the U.S. government had, their own priorities, their own strategies, their own focus areas. We put together the Medical Countermeasure Task Force, which brought in stakeholders from, DOD, DARPA, JPO, JSTO. They brought in FDA, CDC, NIH, BARDA, USDA, DHS, it was really kind of all the stakeholders that, had equities in the space. And, it allowed us to coordinate activities, identify, kind of opportunities for collaboration, do landscape analysis for, for the investments that we should make. It led to the recommendation to invest in a portfolio of medical countermeasures, and it served as the basis of the Operation Warp Speed in May of 2020. So again, it's that collaborative spirit, that collaborative effort where everybody said, okay, we're gonna, we gotta pause. We still have a day job, but we're gonna do that, in between. But everybody came together right away, put in 16, 18, 20 hours a day, 7 days a week, and really helped to focus on that response in the early, and still to this day.
And Rob, I guess you would say that the capability demonstrations
Yeah,
are our war games?
for sure. I mean, I think that I'm super, look, the prototype pathogen I think is step one, but I think if we don't take opportunity when we're doing some of those to put those in a war game like sort of scenario, we did several what I would call mini capabilities demonstrations in 2018 and early 2018 through end of 2018, but in 2019 we really did one that was serious enough that the lead scientist at the end of we did it for Zika, he came to me and said, we accomplished all of our goals. He said, that was really great. Don't ever make me do that again. and we did make him do it again just a year later. But I really think it pushed us to take a platform that we believed in and find all the edge cases, all the complexities. You said this, it's just not, you can think about it, but it's just much more complicated in reality. And if we're not practicing to make progress, we're not going to be able to respond as good as we could.
Great. Well, I really appreciate all your thoughts and thinking about how we can better respond to pandemics of the future. And we're excited to engage with the audience after the panel. So thank you, everybody.